Welcome students. My name is Miss Karen Lenisha Cabral. I'm Assistant Professor at St. Joseph Vaz College, Kottali, Goa. This lecture is for Bachelor of Science, second year, subject Botany, semester 4, course code BOC 104, course title Plant Physiology. Title of the unit Enzymes, module name Structure of Enzymes, outline Introduction, Structure of Enzymes, Holoenzymes, Coenzyme and Apoenzyme. Learning outcomes: describes the structure of enzymes, defines holoenzyme, coenzyme and apoenzyme. Coming to introduction, what are enzymes? Now when we say enzymes, they are biological catalysts which activate various biochemical reactions of a living cell, and it is highly specific and precise in manner. Because of enzymes, the rate of reaction is faster. Without enzymes, this rate of reaction would have been at a much slower rate, and we cannot imagine how life would have been then. Now the term enzyme was coined by Kuhne in 1878. Now all enzymes are proteins and may also contain non-protein parts which could be simple and conjugate. Now Buchner discovered that yeast extract could bring about fermentation of grape juice, just like the living yeast cell. Now what he observed was this extract that had the ability to carry out fermentation would lose its catalytic activity when it was boiled, which means he realized that there was something that was getting, it lost its property, which means it was sensitive to high temperature. Now he coined the word zymase for the active principle involved in fermentation. The substance on which the enzyme acts is called as substrate, and the specific region of the enzyme which is involved in this biochemical reaction is termed as an active site. It is at this active site where the enzyme combines with its substrate to bring about a biochemical reaction. Now we will look at it, where exactly is the active site and what happens to it. Now they have more than one active site, or some enzymes could have just one active site. Now coming to the type of enzymes, that is holoenzyme and apoenzyme together with its cofactor, it is a complete and catalytic activity. Now most cofactors are not covalently bound but instead are highly bound. However, organic prosthetic groups such as iron and vitamin can be covalently bound. Now example of holoenzymes include DNA polymerase and RNA polymerase, which contain multiple protein subunits. Now this is an example of what exactly is an holoenzyme. Holoenzyme, it is an apoenzyme with a cofactor is meant by holoenzyme. So it is a complete structure with a cofactor. Next, coming to coenzymes, they are organic molecules that are non-proteins and mostly derivative of vitamin, which are soluble in water by phosphorylation. They bind apoenzyme protein molecule to produce active holoenzymes. Now coming to the last one, that is apoenzyme, an enzyme that requires a cofactor which does not have one bound. Now an apoenzyme is an inactive enzyme. Activation of enzyme occurs only upon binding of this cofactor, which could be organic or inorganic. Now this is what is an apoenzyme. Without a cofactor, it is called as an apoenzyme. Coming to structure of enzymes, now structure of enzyme can be classified, or it can be categorized in three ways. That is, you have the primary structure, secondary structure and tertiary structure. Now primary structure, enzymes are made up of amino acids which are linked together via amide, that is the peptide bond in a linear chain. This is the primary structure. Now the resulting amino acid chain is called as polypeptide or protein, and the sequence information contains the order of amino acids in the protein starting from the nitrogen terminal to the carbon terminal. Now if you look at the primary structure, that over here it is a chain of this amino acid, the long chain. Now The small section of this image has been magnified, wherein you can see amino acids which are bound to one another with the help of this peptide bond. Since there are many amino acids coming together, it is called as a polypeptide chain. Then coming to secondary structure, the hydrogen in the amino group and the oxygen in the carboxyl group of each amino acid are bound to each other by means of hydrogen bond. This is why it is called as a secondary structure, because of the hydrogen bond between the amino and the carboxyl group. As a result, the protein chains can fold up on itself, and it can fold up in two ways, that is resulting in two secondary structures. One is called as the alpha helix and beta sheet. And these are the images of alpha helix and beta sheets. We will just look into each of this. Alpha helix is a helical structure which is coiled around an axis, and the helix, it is right-handed in nature. It is characterized by intramolecular hydrogen bond between oxygen atoms between the carbon and the nitrogen group of the peptide bond. Now the helix has about 3.6 amino acid per turn on an average, which means in 10 turns it has 36 amino acids bound. Coming to beta sheets, which is the second form of secondary structure in enzymes. Now it is a beta plated sheet. It is formed by intermolecular hydrogen bond between two or more straight chains. That is the oxygen atom of the carbon and oxygen of peptide bond on one strand, and hydrogen bond with NH group on the peptide bond on the adjacent strand. Now since there are two forms, so it is called as parallel and anti-parallel. Now if the amino acid of both chains are on the same side, then the sheet is said to be parallel. If the amino group of both chains are on opposite side, the chains are said to be anti-parallel. Now the anti-parallel beta sheet is more stable than parallel sheet, owing greater alignment in the hydrogen bond. Now here you have the anti-parallel bond because they are on the opposite side, whereas the second image is a parallel beta sheet. The tertiary structure, as a consequence of folding up of the 2D linear chain in the secondary structure, the protein can fold up further, and it gives a three-dimensional structure which is called as a tertiary structure. Now tertiary structure is defined by the atomic coordinates, and it is stabilized by both covalent as well as non-covalent bonds. Now the image is put over here, which is a 3D image. Now we will just quickly go through the binding sites of the enzyme. It has 5 binding sites. The first one, the substrate binding site, which is the areas of an enzyme where the substrate binding occurs. Now second one is the catalytic site, one or many sites located near the binding site, and they perform the catalysis, that is, it accelerates the rate of reaction. And thirdly is the active site. Binding site and catalytic site together are called as the active site. Now cofactor site and allosteric site are the additional sites for binding of cofactors and allosteric moderators. So a protein structure appears this way, wherein the blue color that you see is the binding site, whereas the red is the catalytic site, and both together are called as the active site. These are the references. Thank you.